So I'm very, very excited for our speaker today. Maggie was sitting in your seat not that long ago. She did her master's degree with Anne McElroy in Boston alum, where she was looking at antimicrobial stuff in the shells of lobsters. Since then, she went on and got her PhD at the Okinawa Institute of Science and Technology. She was also a Simons postdoctoral fellow and went to Woods Hole Oceanographic Institute. And now she's an assistant professor at the University of South Florida, where she has started the MICO Lab. I had to write it down though. Microbial interactions in a changing world, where she's really addressing three questions. What are the mechanisms for interactions between microbes? How did they evolve? And how does their breakdown affect things, the ecology of the system? And so I'm very excited for Maggie giving her talk today. Let's welcome her.
They warned me about the lights going off. That was still a little startling. Okay.
Did you turn them on?
Yep. So, thank you. I'm not loud though. Is it for, am I supposed to be loud?
I think
Just a little.
I think it's okay back there.
Pretty soft.
The
Okay.
Speakers are back.
But I mean, it doesn't sound like the microphone is making, it's just for the computer.
No,
it's-
they
it's-
hear it, it's just in the back.
Okay,
Yeah.
great. Sorry, thank you. Okay, so today I'm going to be talking about interactions between phytoplankton and bacteria. And this has been one of my big passions in my research career. Brad kind of okay.
Is it working now? You go back and forth for a second.
Yeah.
There you go. Sorry about
So Brad a little bit stole my thunder with what I wanted to say at the beginning. So I wanted to start out and kind of say a little bit about my science path and why I'm so excited to be here today. So I actually started my interaction with Stony Brook as a high school student. I was in the high school WISE program, and at that time, I don't know how the program has evolved over time, but we would come to campus one day a month throughout our entire high school career. And so that really was a huge part of what got me into science and thinking about research as a career. And then as thinking about undergraduate work, because I had spent so much time on the Stony Brook campus by that point, felt a little bit too close to home for college. So I went to St. John's for undergrad, but then I came back for my master's degree. And that was like a really special time for me in my life. I met my husband here, made really great friends. And basically, I'm always thinking about SoMAS and how I can recreate some of the magic of SoMAS in other places that I work. So I did my PhD in Japan and postdoctorally. Now I've landed and I'm starting my own lab at the University of South Florida. And I was telling Brad at the beginning of the day that I am truly trying to recreate SoMAS in my department now. So I'm working very hard to try to get a Vax to Flax up and running. This is like my passion project. Also trying to get Friday beers going. I don't know if you guys still do that, but lots of really great community things that you have here don't exist everywhere. So I guess just like, this is a really special place and you're all really lucky to be here and I'm jealous. So jumping into the science part, I think everybody here knows that phytoplankton are really key to biogeochemical cycles in the ocean. But all of these interactions between phytoplankton and bacteria and other organisms are happening at these really spatially structured but small scales. And then these very, very tiny scales are scaling up to these giant impacts that have global ramifications. So the phycosphere is the word that is used now to describe the region around phytoplankton, kind of like the rhizosphere is the region around roots. And there's all sorts of different interactions that happen in this region. And they have implications for productivity. So bacteria will produce different metabolites that the phytoplankton can use to increase growth. Sometimes the bacteria can stimulate toxicity among algae. Obviously, bacteria are really important in the microbial loop, so recycling nutrients. They also can stimulate just regular phytoplankton blooms, not harmful algal blooms. And then finally, it can have impacts on climate. So a lot of phytoplankton will produce DMSP, which then is broken down to DMS and usually acrylate. That DMS is a climatically active gas. So the bacteria are chemotactically attracted to the DMS or DMSP, and then they help break the DMSP into DMS and acrylate, which then can cause more cloud formation and actually have cooling effects. So these interactions are very tiny, but very big and important in other ways. So some of these different interactions between phytoplankton occur with across the all the domains of life. So we have protistan grazers and parasites that are interacting with phytoplankton. We have protists that act as hosts or cytoklepti or where they steal the whole cell, or sometimes they just steal the plastid. We also have symbiotic bacteria that live inside phytoplankton, symbiotic bacteria that live on top of phytoplankton, and we have opportunistic bacteria that can sometimes be friends and sometimes be a little bit not friends, depending on what the situation is outside of the relationship. So throughout most of my career, I've been focusing on one main group of phytoplankton to think about all of these different interactions. And that group of phytoplankton is Phaeocystis. So I'm going to go into some details about why Phaeocystis is such a special phytoplankton, especially when we're thinking about these interactions. So my first part of my talk is going to be thinking about Phaeocystis, and then I'm going to talk about some other phytoplankton that I've been working on more recently. So Phaeocystis is a globally abundant bloom-forming phytoplankton genus that is especially interactive. So it can be a single-celled organism, just kind of swimming around in the flagellate stage. It has this really amazing colonial stage where you have these large mucilaginous colonies with lots of individual cells embedded inside of the colony. And Phaeocystis is also an endosymbiont of other protists, so they can live inside of acantharians, which are really interesting in themselves. They have strontium sulfate skeletons. They're really heavy, so they can have really interesting impacts on biogeochemical cycles as well. There are a couple different species of Phaeocystis out there that we know of. There's also some that are kind of genetically implicated from big metagenomics surveys, but we haven't cultured or talked about more deeply. So some of the main groups are Phaeocystis puchetti that lives kind of in the Arctic area, Phaeocystis globosa, it's the whole middle of the planet, and then Phaeocystis antarctica, near Antarctica, Southern Ocean. And then we have some species that are warm water dwellers, so Phaeocystis cordata and gani. I'm not going to talk about them very much today. We don't know that much about them. They're mostly flagellates. Sometimes they maybe make some colonial weird things, but they are potentially less interesting for some of the things that I'll be talking about today. So when Phaeocystis is blooming, they're making these giant colonies, and those generally occur in the English Channel, in the North Sea. There's been some Phaeocystis blooms along the Atlantic seaboard in the United States. There's also been some kind of more harmful Phaeocystis blooms, near Vietnam. And then there's giant Phaeocystis blooms that are a really big, important part of the ecosystem in the Southern Ocean. And then Phaeocystis, when it blooms, it can really, really bloom. So it has really big effects. One of the things that happens when you have a big Phaeocystis bloom is these foam events. So the colonies have lots of TEP or EPS, lots of polysaccharides, and when it's very windy, all of this gets whipped up and it produces these large foam events. So a couple of years ago, 5 years ago now in 2020, there was one of these very, very large Phaeocystis events in the North Sea that actually led to some of the first human deaths due to Phaeocystis blooms, where people that were normally very proficient in being out in the water were stuck in this foam and actually ended up drowning. So even though Phaeocystis is a normal and important part of the ecosystem, if it's becoming more abundant and the blooms are getting more aggressive, it can have some negative impacts on ecosystems and people. And when these very big blooms happen, they often turn out to be very large export events. So Phaeocystis colonies, because of the makeup of the colony, they're kind of unpalatable zooplankton, and there's also a size mismatch. So they're a nanoflagellate, but they're in a colony. So what would normally eat that type of organism can't eat them because they have this large gelatinous colony around them. And so I'm going to show this video, hopefully. Yes. Long title. And then we have little nuclei. There's a Phytoplankton colony. Nope, I don't want that. And this is one of my favorite little videos I've found. There's actually from a study they tied lots of different stages of copepods. Well, actually, they glued them to strands of hair, they said, and then they videotaped them interacting with a whole bunch of different phytoplankton types and assessed the importance of colony formation, whether it's a Phytoplankton colony or diatom chain on prey avoidance. And it turns out that a lot of these little predators, they do like diatom chains, which I thought was very interesting. They eat them like little corn cobs, but the Phytoplankton gets turned around, looked at, and thrown out. So that's part of the reason why Phytoplankton will be exported often. Also, these colonies will get very heavy, so when they start to senesce, all the cells kind of detach from the colony, and they fall down to the bottom of the colony. That makes them kind of heavy, and they fall down. So these are some pictures and plots from a more recent-ish paper that's a little bit local. This is from the Martha's Vineyard time series, so just a little bit north of here. And there was a very, very large Phytoplankton Bacterial bloom that occurred in the spring. And so this is underwater vision profiler images, and this is some IFCB images. And they followed this bloom over time, and essentially they saw all of it sink and then move out over the shelf and then down into deeper water. So all of this biomass was not being consumed, it was being exported out into deep water over the shelf. So in my work so far, I have spent a lot of time thinking about how Phytoplankton interacts with photosymbiotic hosts, parasitic protists, symbiotic and opportunistic bacteria. And today I'm going to focus more on the interactions between Phytoplankton and the symbiotic and opportunistic bacteria. And then later in the talk, I'm going to come back. Oh, sorry, I keep getting ahead of myself, sorry. In each of those individual types of interactions, this colony formation is an important part of why those interactions work. So when it's in this photosymbiotic stage, where it's inside of the acantharium, kind of its tendency to live in that colony and to put stuff out into the colony and then take organic material back from the colony into the cells makes it very well adapted to live inside of a host, where it's basically doing the same thing. It's sharing material to the host and then taking material back from the host. The colony also kind of primes it for bacterial interactions because the colony is primarily polysaccharide, which bacteria really like. And there's also some chemicals in the colony that attract bacteria. And then finally, this interaction I'm not going to talk about very much today, but this is a very cool interaction where a dinoflagellate goes inside of the Phytoplankton colony and sucks out all the plastids from all of the Phytoplankton cells inside of the colony. And then it leaves the Phytoplankton colony and it leaves a ghost colony. And that empty colony ends up being exported. So all of that carbon then gets exported. So bacterial interactions with phytoplankton is kind of like a hot topic, like microbiome stuff. There's lots and lots of work between thinking about how diatoms and bacteria interact. Also coccolithophores, they're a really great model organism. So we have all of this information that bacteria are providing growth factors, like the same types of kind of plant hormones that you learn about in high school biology that cause plants to grow in certain directions. Bacteria give those things to phytoplankton to increase their growth. Bacteria give vitamins to diatoms, they give specific amino acids, and then there's different signaling molecules that are triggering cell death sometimes, or cell division. The diatoms are mainly giving organic carbon back to the bacteria. So we know all those things are happening. We also know that diatoms and Phytoplankton are often in direct competition. So diatoms or Phytoplankton will bloom first or second in different spring scenarios. And sometimes diatoms and Phytoplankton will be exclusionary. So one will bloom one year and the other will bloom another year, but they don't bloom at the same time. But until I started looking a couple years ago, we didn't know anything about bacterial interactions with Phaeocystis. So I'm going to spend some time today talking about those interactions. And so these are all of the kind of proposed ideas about what the colony might be giving to bacteria. So there's lots of complex carbohydrates in the colony. There's a lot of organic nitrogen compounds, a lot of DMSP, and acrylate. And this is a picture I actually took last week. So we have a Phaeocystis colony there, and you can see, hopefully, there's like a, from where you are, there's like a halo of bacteria around the colony. So this is an example of that chemotaxis with lots of bacteria kind of coming towards the colony and hanging out in that phytosphere region around the Phaeocystis colony. So at the end of the talk, I'm going to come back to thinking more broadly about phytoplankton, and then I'm going to talk about another phytoplankton that has newly come into my life. Phaeocystis has been the center of my life for many years. And now that I've moved to Florida, I have some graduate students, pyridinium has become second child that's trying to take the spotlight from Phaeocystis. And this is also going to be the first time that I'm presenting student work. So it's kind of an exciting new thing to do. I'm going to do my best to do a good job for my graduate student, Lydia, whose project this is. So starting with the phaeosphere, some major questions that I had is, first, Phaeocystis doesn't have a core microbiome across species, across individuals. And then secondly, what kind of interactions are actually happening there? Are the bacteria being useful, especially when the conditions might get a little bit unfavorable for growth? Oh, okay. There's a map here. I think that the color is just so light, maybe it's not coming, it's a gray map.
Yeah, I get the Zoomers can see it.
Okay, great. So essentially I had 4 Phaeocystis strains. They were originally isolated from 4 different places across the planet. And they were isolated at very different times in history. So some of them are very old. CCMP 629 was isolated all the way back in 1982. It's older than I am. And there are, then there's a range, 1992 and 2006. And then the last strain from the Kuroshio current was one that I isolated. And that's mostly important because that Kuroshio strain never lived at Bigelow. The other 3 strains lived in Maine together, not in the same vial, but in the same incubator for many years. So that could influence kind of the questions that I'm asking. So I grew all of these different strains over time. And then when they're in the same growth phase, I isolated individual colonies from each of these replicates of each strain. And then I did single colony DNA extractions, and I amplified the 16S rRNA gene region to look at the microbial communities that were associated with just the individual colonies. And then I also looked at the bacterial communities that were in the background media. And then the other part of the study, I thought about whether or not, if we take away all the bacteria that are there already, if we treat them and make them azenic, and then we give them the opportunity to recruit new microbiomes, do all of these different strains recruit the same bacteria again? Or is it more stochastic in the beginning, and then it takes a long time for it to kind of coalesce into this common community? So treated them, let them have some natural seawater, and then grew them for a couple of culture generations, and then kind of redid that same thing where I isolated individual colonies and did sequencing for the individual colonies. And the first thing that popped out is that so the kind of light blue that's on top here is just the media communities. And then richness is the total number of species of bacteria that are present. And so I'm looking at how many different types of bacteria are in the media versus on the colonies. And there's about half as many different bacteria on the colonies as there are in the media. So the things that are actually physically associated with the colony are a subset of what's available in the media and the background. And then in the recruitment trial, we got basically the same numbers, where double the amount of bacteria were recruited into the media, but half that amount was recruited onto the physical colonies. And then the amount of bacteria that was in that filtered seawater was a lot higher. So when we did that recruitment, it was very selective. It went from 400 to 40 that were kept over time in the media. And this is the principal PCOA plot, actually, of the Atchison distance between communities. So that's a statistical method to kind of diminish the impact of compositional bias, which is a problem with this type of data. And so in blue is the recruitment trial, and the triangles here is that original seawater. And then in yellow is the original communities. The squares are the media, and then the circles are the individual colony communities. And you can see that the individual colony communities, whether it's the original or the recruited community, they kind of converge together. If I didn't put a color there, that's really like one blob. Even though there is some difference between those two trials. But the stuff that's in that background media, that's a little bit more separate, a little bit more different. And then the seawater is kind of all the way over there in the corner. So this plot is like really big and there's lots of stuff here, but it's actually one of my favorite plots. I call it the magic carpet plot. So on the left side are individual ASBs. So these are like very granular levels of diversity, and I have them grouped into orders. And then across each bubble, each column is one individual colony or one individual media replicate. And the bubble size represents the abundance in that individual sample. So what I want you to see from this plot is that there's individual ASBs that are present in all of these different communities. And when there isn't the same ASB present, there's something that's closely related that is kind of like making up that missing spot. And some of the main groups that were really important in these communities were Alterramonidalis groups, Burkholderialis and Rhizobialis. And these are groups that are previously known to be part of phycospheres and rhizospheres that have genetic potential to produce plant growth hormones, B vitamins, and siderophores. So this is just metabarcoding. I don't know if these specific bacteria can do all of these things, but their friends and relatives are able to do those things, which suggests that they might be doing this type of thing for the Phaeocystis colonies. So since the communities were so consistent and reproducible, I thought there must be a reason, they must be doing something. So I decided to test how the azenic versus the xenic Phaeocystis did if I took away their nitrate resources or their B vitamin resources. And I chose these two things because these are kind of potential things that from the literature are likely candidates for the bacteria to be doing for the phytoplankton. So first, when we take away nitrates, the Phaeocystis cultures that have no bacteria with them are able to live much longer. So this suggests that when nitrate is limiting, the bacteria are competing with the Phaeocystis for this resource. So the black is the azenic, and the triangles with the empty is the non-azenic. And we can see over time that the triangles drop out, but the circles keep growing even though there's not nitrate being provided. And this is particularly because Phaeocystis stores a lot of nitrogen in that colony. So if there's not being competed for, they're able to grow over time even without nitrate added to the media. And so this is the same thing now without B vitamins added, and we have the opposite pattern. So when we take away B vitamins, the Phaeocystis that have bacteria still in their cultures, they're able to keep living, whereas the ones without bacteria can't keep living. And then this is the control. They just get everything, and they both just keep living because they have everything they need. So some take-homes from this first story that I wanted to share with you are that Phaeocystis globosa bacterial communities are consistent across strains, even though these strains come from very different places and have been in culture for very different amounts of time. And And this I just want to point out is different than has been seen for other phytoplankton, so that's why it's kind of interesting. So like coccolithophores, there's tons of EHUCs in different culture communities. And when you look at all of those different EHUCs, their bacterial communities actually represent where they were isolated from. So they all have different communities, whereas the Phaeocystis has this one community that's consistent. And if you take away their bacteria, but you give them the chance to get new bacteria, then they all get the new bacteria that's the same. And then this bacterial community can either be a friend or a foe, depending on what the environmental conditions are at that particular time. So what about other Phaeocystis species? I had the opportunity to go to the Southern Ocean, so I turned my attention to Phaeocystis antarctica to see if Phaeocystis antarctica likewise has this very consistent community, bacterial community associated with it. And this was super exciting for me because it was my first time really being in a Phaeocystis bloom. You know, we see all of these amazing pictures of the foam. We talk to other people that have gone to the Southern Ocean and they complain rampantly about the stink of the Phaeocystis on the boat. And then I got to go and it was there and it smelled so bad. And actually it kind of like invaded our water maker. So all of the fresh water on the ship smelled like sulfur, and even like the water you're supposed to wash your face and brush your teeth with, it's just pure sulfur. Everyone on the boat was furious except for me. I was so excited. So this is the track for the Palmer LTER grid, the year that we went, and kind of offshore we had this large phytoplankton bloom, and as I'll show you, this was about 50% Phaeocystis. And when we were out in this like really high biomass bloom area, it was amazing. You could just take a jar of water and hold it up to the light and you just see Phaeocystis colonies. So I would just put it on the microscope, isolate individual colonies, wash them as much as I could. So that's an example of that. I have one colony in one drop of water by itself. And then I would take that one individual colony and I did DNA extractions on the boat, actually did PCR on the boat, made sure all of my PCR was working, so that now instead of having this culture of potential bias, I'm looking at the bacterial communities that are really, really, truly associated with Phaeocystis in the environment. There's no way that I could get closer. They're taken right out of the ocean. So I had 11 colonies from the 400 station and 15 from the 600 station. And that's the cute little instrument that I use to do my molecular work on the boat. It's called a bento box. It's very fun and easy to use. I highly recommend. And throughout there's no mouse here. Throughout the rest of the grid, I have background samples. So looking at what are the phytoplankton and the bacterial communities in the water, not just on the Phaeocystis colonies. Yes, very white. And then the other part of this was I was really worried about that culture bias from my first study. So I wanted to, in this case, see how the community changed in culture over time. So I took some of these colonies that I was isolating and I made culture from them. On the ship, they have really nice culture facilities. I carried them with me on the airplane very carefully, and then I kept them in culture for a year, and then I redid this whole thing, sequencing the bacterial communities from the individual colonies. So these are the communities that I found on the individual colonies from the fields. This is by order, and what you can mainly see here is there's a little bit more variability than there was in those cultured communities, but essentially we have the same overall community on each of those individual colonies that were isolated from two different places. And some of the main contributors, again, we have Altera monodalis. Here we have Oceanospiralalis. And then there's a single monodalis at the bottom. And then there's colors are all different. Hold on. And this light blue one is the Colobacterialis, which is also an important one. So this is another look at the ASD levels. So this is just Alteramonas species or genus, or they're ASDs, and then they're grouped into genera. So one thing that was very interesting is that there was one ASD from this Paracleocola that was present in every single colony and made up close to 50% of the community in all of the different colonies. And then we also had a lot of Colloquium that was present in all of the different colonies, kind of making up this core community. These are both psychrophiles. They like really cold temperatures, and they've been found previously with ice algae, and they've been associated with, from environmental samples, with decaying Phaeocystis blooms. Another interesting here is we have an interesting one here is Marinobacter. So Marinobacters, when they're co-cultured with diatoms, they actually stimulate the diatoms to produce more polysaccharides. And as a result, they increase aggregate formation, and they cause those aggregates to sink faster. So the presence of this bacteria can increase export of just phytoplankton blooms in general, but maybe in this case, Phaeocystis blooms too. So these are the results from the CTD and the underway samples. So this is looking not at the individual colonies, but at the kind of larger community. And we have this same pattern. So the Western Antarctic Peninsula is here. And as we move offshore, similar to the chlorophyll that I showed at the beginning of this, we're having an increased proportion of Phaeocystis further offshore, but we also have an increased proportion of diatoms further offshore. So that's a little bit conflated. And nearer to shore, we have more of those smaller nanoflagellates like cryptophytes and firstenophytes. And then we have a similar pattern in community dynamics for the bacteria. So when we get closer to shore, I think the starkest pattern is that we have a lot more SAR11 when we're closer to shore, when there's more of those firstenophytes and cryptophytes. And we have more Flavorbacterialis and other things when we're offshore, when there's a higher proportion of phytoplankton, probably more organic carbon available. So we have more of those copiotropes that really like that high nutrient area. So I also tried to correlate these different bacterial groups in space with the presence of different phytoplankton. And there are some direct correlations between Phaeocystis abundance and specific bacterial groups. And these are some of the groups that I already pointed out on the other plot. And if you look at them this way, for example, the Paraglyciacola and SAR92, which is a very cool story, are directly correlated with Phaeocystis abundance. SAR92 is interesting because it's one of the bacteria that are very attracted to DMSP. They have the pathway to break down DMSP to DMS and acrylate, and they are often there's only one other study before this that has looked at bacteria associated with Phaeocystis antarctica blooms, and SAR92 is what they pointed out. They're like, this is more abundant when Phaeocystis is more abundant, and it's not super abundant in my samples, but it is correlated to Phaeocystis abundance. So it's kind of reiterating that previous study that probably that was true. So what about when we put them in culture? So have those guys in culture for a year. I guess, probably a lot of you have been to Woods Hole before. Some of the 4-degree rooms where things are grown, very old, and are not necessarily 4 degrees all the time. So I would say that this 4-degree room was like 4 to 9 degrees. And one thing that was very clear in this culture situation is one is that diversity decreased over time. So it lost about half of the diversity in those microbiomes after a year of culture. And the community structure changed significantly. And the main thing that happened was that those psychrophiles dropped out. So those organisms that were the main component of the Phaeocystis communities, when I kept them in culture, those cold-loving organisms died. And other organisms became more abundant, particularly Sulfitobacter. And this one I think is important to think about and to be a little careful with, because Sulfitobacter is widely reported as being a very, very important component of phycosphere communities, but it might just do really well in culture. So in my culture conditions, it was the most abundant organism that was associated with the Phaeocystis colonies. So thinking about that going forward, Sulfitobacter might be helpful to phytoplankton, but it might also just really like living in culture conditions. So some take homes from this part, Phaeocystis antarctica colony microbiomes are also very consistent across time and space, or sorry, across space and colonies, but they're different than the Phaeocystis globosa microbiomes. In the field, the Phaeocystis antarctica co-varies with major microbiome constituents, including bacteria that are important to the sulfur cycle, and psychrophilic bacteria turns out don't do well when you have them in a kind of crappy 4-degree room in the basement of the building. And I think that this is kind of necessitates, especially when we're working in more extreme environments that are challenging to replicate in the field. So I have like the temperature is a little bit of a problem, but also the Southern Ocean is very different than a bottle in a basement, right? So there's a lot more turbulence, there's ice, there's all these different things that you can't recreate in the culture conditions. So if you're really interested in these more extreme, harder to replicate environments, it's probably pretty important to go there. So I want to switch now from talking about Phaeocystis and think about Pyridinium. So Pyridinium is a very different organism. My graduate student Lydia was really excited about Pyridinium when she joined my lab because she had been working at the Florida Fish and Wildlife Research Institute before that, and had already been thinking a little bit about Pyridinium blooms in Tampa Bay. So when she joined the lab, she's like, yeah, I really want to work at Pyridinium. And I was like, okay, well, what about bacterial interactions with Pyridinium? And so that's the direction that we've been working on. So now we're going to be exploring the pyrosphere. And it's just very convenient that it also starts with a P and has an O. So we got the phycosphere, the phaeosphere, and the pyrosphere. So Pyridinium is an emerging HAB species in Florida. If you think about Florida and HABs, probably the first thing that comes to mind is Karenia brevis, the big red tide organism. Karenia brevis has been in Florida Gulf Coast waters forever, for as long as people have been reporting conditions in the region. They have been telling stories about fish kills and red tides. Pyridinium is different because it really hasn't started blooming in the region until 2002. So around 2002, these blooms started to become annual in both the Old Tampa Bay estuary and on the other side of the state, the Indian River Lagoon estuary. And over these 20 years, where people have been monitoring and researching the Pyridinium blooms, the usual suspects, temperature, salinity, nitrogen loading, these are not directly correlated with cell counts. So when macronutrients kind of fail to reliably forecast phytoplankton growth, oftentimes micronutrients, trace metals, vitamins, maybe some organic matter are having an important role regulating the growth of these organisms. We also know that most HAB dinoflagellates do have a B vitamin requirement, and some HAB dinoflagellates rely on phycosphere bacteria for at least their B12 quota. So Lydia's hypothesis was that vitamin B12 availability and bacterial interactions are playing a role in Pyridinium bahamen's bloom formation in Florida estuaries. And our objectives were to first determine Pyro's B12 requirements, determine if they're able to recruit specific communities of bacteria, and then determine if those recruited communities are able to actually supplement B12 if B12 is not available for them. So we tested Pyro's ability to grow if we take away B12, biotin, and thiamine. These are kind of the 3 B vitamins that phytoplankton most often need. So we grew them in these 3 media, missing 1 B vitamin at a time, and then we also grew them in replete media, and Lydia measured their growth every 48 hours or 4 culture generations. I have to note that Pyro grows a lot slower than Phaeocystis, so my culture generations were 5 days long, and Lydia's culture generations were 30 days long. So this is a lot more work for her than it was for me when I was doing it myself. So these are the results from those culture generations, and then so I have to point out that they weren't true culture generations because as soon as we took away B12, the Pyro just died, which I don't even have the results up for the biotin and the thiamine because they don't need it. They can grow forever without biotin and thiamine, which is different than Karenia. Karenia needs all 3 to grow. So the for each of these generations, we just started the minus B12 again. So these are really more replicates showing you that every time that we do it, if we take away the B12, they just immediately die. This is a little bit unique. Usually it takes at least one generation for them to use up any luxury storage that they have. So they have probably, we'll see, a pretty high quota. So our next question then was, well, how much do they require? So we did a 10% dilution of B12, and it looks like there's so there's kind of like a switch here in the middle between 3.69 and 36, where we start to have like bloom level biomass here, and we have less than bloom level biomass before. But to have any growth at all, they seem like they can grow at least a little bit if you just give them a tiny bit. But we're going to draw this line in the sand in the middle there. And if we compare that to other bloom forming dinoflagellates, these ones in the middle in the light blue box, these are the ones that also produce saxitoxins, which is the toxin that pyridinium makes. So pyridinium seems to need more than they do in order to reach bloom level biomass. And Carenia brevis, the real bad guy, they need a lot lower quantities of B12. So it seems that pyridinium has a very high B12 quota. So something I didn't mention yet is that we didn't do anything to these pyridinium to remove their bacteria before we started this experiment. Essentially, I was like, Lydia, just like try some growth curves, see what happens. Probably it's not going to make any difference in the world because they are probably full of bacteria, but we'll figure out how to make them azenic later because that's a lot of work and it takes time. So just try it. Let's see what happens. And then this happened, right? We took away their B12 and they just died. So we were kind of shocked and surprised, and we're like, what? Why? So we just did an initial test where we put the old Tampa Bay isolate in marine broth, or like a tryptone marine broth that had a matching salinity to the local estuaries, and they were always negative. They always tested negative for no bacteria in the cultures. And then we have this other isolate of pyridinium from the other side of Florida, from Indian River Lagoon. And when we use these two test media with the Indian River Lagoon, it's like positive immediately, lots of growth. So there's something different between these two isolates. It turns out that it probably has to do with how they were originally isolated, or really how they were maintained at FWC. So they were both isolated as cysts, which are resting stages that are in the sediment. The Indian River Lagoon isolate was isolated in 2016, and the old Tampa Bay isolate was isolated in 2023. Before 2020, they did not autoclave their seawater for culturing. After 2020, they autoclaved their seawater for culturing. So the Indian River Lagoon probably was pretty close to sterile when they originally isolated it, but over time, when it was grown in non-autoclaved seawater, they got some bacteria back, whereas the old Tampa Bay isolate stayed very clean over time. It was fresher, it was newer, and it had been only grown in autoclaved seawater. So we then did some filtering in order to, and sequencing to see what these communities actually looked like. And what we found is that the Indian River Lagoon isolate, the one that had been in the non-autoclaved seawater, it has much higher diversity, whether it's in the free living stage or in the particle associated stage. But we have this pattern that's kind of similar to what I talked about on the Phaeocystis, where the actual particle associated communities are lower diversity than the background communities. And then the old Tampa Bay is like, there's no diversity in that community, and there's no difference between the particle associated and the background. And in those bar plots, you can see that the old Tampa Bay, that's the one on the right, OTB, it's like the lowest diversity community I've ever sequenced. There's 9 ASVs in this community. And these 9 ASVs were from sediment associated and rhizosphere associated bacteria. So stuff that probably was maybe on the cyst when it was isolated, or just kind of around in the sediment when it was isolated, but not normal phycosphere or seawater communities at all. So we kind of like accidentally stumbled into this great opportunity to be able to test what's going on with this organism, because it's so hard to make azenic cultures, and we just kind of got like an almost azenic culture for free. So the next thing that we did is we let these almost azenic culture recruit new bacterial communities from the IRL culture, from the Old Tampa Bay surface seawater, and from the Indian River Lagoon surface seawater. And then we sequenced it again to see if it worked. So now, compared to before, we have pretty high diversity communities associated with the culture. And we have a difference between the particle associated and the free living, the free living communities. So essentially it works. There's new communities in there and maybe they'll be useful. And then I just want to point out, very different than the initial. So then we tested these different cultures. We challenged them to low B12 conditions. The Indian River Lagoon culture bacteria, no help, still dead immediately if you take away the B12. The Old Tampa Bay surface seawater, also no help, dead almost immediately when you take away B12. But the Indian River Lagoon water, there's a little bump. They got to grow a little bit. They're not dead immediately. So this is potentially promising. We haven't actually sequenced the Indian River Lagoon recruited communities at the time of putting this together yet, but that's happening currently. So that part of the story, I'll be able to talk about sometime in the future. So that's our immediate next step, is we can finish up all that sequencing to see what's different between that Indian River Lagoon seawater community. And then because we have these other communities, they can kind of like give us a subtraction point. So what is in this community but not in the other ones? And we can start targeting those bacteria to see what they're capable of and what they're actually doing to see if they really are providing B12 for these phytoplankton. So our takeaways is that pyridinium is a B12 auxotroph, but like a hardcore B12 auxotroph. Indian River Lagoon culture and seawater, OTB seawater is not good enough, but Indian River Lagoon seawater bacteria might provide enough B12 for pyridinium to grow, but probably maybe not enough for it to support full bloom biomass. So maybe there's some other sources that are needed in order for it to really bloom the way it does in the summers in Old Tampa Bay and Indian River Lagoon. So in summary, Phaeocystis has really specific colony microbiomes. These interactions are really important to let Phaeocystis outcompete other plankton groups and form these really high biomass, high carbon export events. And the specific bacteria that are there are important to bloom demise, export, and carbon cycling. And the pyrosphere bacteria might influence bloom dynamics, but it maybe doesn't seem like B vitamins are a major currency of exchange, at least with the experiments that we've done so far. So with that, I'm just going to say thank you to everybody that's contributed, especially Lydia, who did all the experiments for this third part. And hopefully I can take a couple questions.
We have two students that want to ask the first two questions.
Great.
Ian and Sebastian.
Yeah, that's the first one. So it seems a common trend between the relationships between algae and bacteria are those that have mucilage, whether it be EPS or other polysaccharides. Has there been any methods looking into controlling the polysaccharides to control bloom? Because
So I don't, that's probably very hard to do.
Yeah.
But there are some very cool experiments out there that are kind of starting to go in that direction. So Justin Seymour and Roman Stockard have this cool little device where they put individual algal exometabolites or polysaccharides into each well, and then they put that into the ocean for some time, and then they look to see what bacteria are recruited to each individual polysaccharide. So that's starting to kind of have more specific connections between individual polysaccharides or exometabolites and specific bacteria. And they've also have some interesting experiments where they use like microfluidics to watch bacteria swimming towards certain things. But truly getting down to the core of your question, like in the field and then scaling that up, I think that's something that we're going to have to keep working on for a while.
Thank you.
Yeah.
second dose and very interesting topic. I want to ask that would there be any additive relationship between the photosynthesis and dietol?
Yeah, that's a really great question. That is something that I'm working on from my postdoc a little bit. So in Cape Cod Bay, Phytocystis puscheti has become a sometimes bloomer, which was not historically the case. It was kind of always like Gulf of Maine, Cape Cod, Massachusetts Bay, diatoms in the spring. Around 2010, we started to get these really big Phytocystis blooms. So I did like a time series study over 3 years where I did metatranscriptomes every month for the whole year for all 3 of those years. And we have like a Phytocystis bloom year and a diatom bloom year. And so we're going to be looking at the transcription of the Phytocystis, the diatoms, and the bacterial communities over time in regards to different environmental conditions to kind of start getting at what you're getting at, like the competition between Phytocystis and diatoms and how the bacteria are playing into that competition. That's a lot of data, and I'm not ready to tell you any answers yet, but it's in the works.
Do you know if any of the toxins produced by, say, some of the toxic dinoflagellates or diatoms, do they have any antibacterial activity? In other words, do they? No one knows why they make this stuff.
Yeah.
And it's expensive for them to make. So I was wondering if it has any antibiotic type properties that keep them from getting sick from bacteria.
Yeah, that's a good question. I don't know for saxitoxins. I don't know the answer if that's antibiotic or antimicrobial at all. For brevetoxins, I don't think that they are. A really recent paper came out. There are several species of bacteria that co-occur with Carnea brevis blooms that actually kill Carnea brevis, and it seems that the Carnea can't do anything about that when they're in co-culture. The bacteria are still algicidal. There is some other kind of interesting interactions to think about, though, in that some culture experiments have shown that bacterial presence does increase toxin production. So whether or not that's because they're trying to kill the bacteria or because the bacteria are like, oh, well, if you produce toxins, then there's going to be more decaying organic matter around. You know, I don't know how that relationship evolved or like what the goal is there. But it's definitely like a very interesting sphere to think about for the future.
We have a question on Zoom from Chris Gobler. Why do you think that the 9 ASV bacteria in the IRL xenic cultures do not synthesize B12?
That's a very good question. Especially because they're rhizobialis, it's usually a B12 producer, and they are previously found in rhizospheres of mangroves and other salt marsh and saline plants. You would expect them to be B12 or B vitamin producers. Don't know why they're not.
Other question?
Mentioned acrylate as sort of a side product of the episode productive, but acrylate could be biologically active too, and I think it can, at least a big pepper in the full paper and big one stomachs that it kill bacteria. Can you say anything about the role of acrylate?
Yeah, 100%. That's one that I've highlighted before specifically because it is antimicrobial. But not against everything. So my thought there is that it might be kind of similar to like the microbial system with the squid, where they describe it as a winnowing, where you start with a larger diversity community, and then there are chemical and mechanical factors that kind of make that bacterial community smaller and smaller and smaller until it's the very specific bacterial community they want. So I don't know that that's true, but like in the stories that I make in my mind, I think that might be part of that.
All right, we have time for one last question.
Hi. So I noticed a lot of your data you presented was done at the order level.
Mhm.
And I was wondering if you could speak on why you decided to choose that, and maybe if you see similar trends, if you go more specific to like maybe family or genus level as well.
Yeah, good observation. The reason that I plot them at order level is because I don't have enough colors to go any deeper.
Especially on that one.
But the actual analyses do go deeper. So I will generally break out per order and then look at all of the ASV levels. And then all of the multivariate statistics are at ASV level. It's just that those bar plots,
Yeah.
it just can't. And the order level will often catch like the major trends, and then I can go into the deeper level of analysis without having it be in color.
If there are no more questions, thank you again for this nice talk.